Hello, my name is Niccolo Bolli. I'm an academic clinical lecturer at the University of Cambridge in the United Kingdom. I'm here at the ASH meeting presenting results of a study looking at heterogeneity of subclonal architecture and mutational profiles in multiple myeloma using next-generation sequencing. In my study, I used next-generation sequencing technologies to look at the genome of multiple myeloma. Multiple myeloma, it's a disease that is nowadays more treatable than before, but still not curable in the vast majority of patients. So we wanted to look at what are the alterations in the genome of multiple myeloma cells and how they change over time. To do so, we used DNA samples from 67 patients, and in 15 of them we had access to multiple samples over time at the time of disease relapse. We sequenced the DNA, or actually the coding portion of the DNA, for all these patients, and we found some very striking features. The first is that every sample is a complex mixture of different subclones, so the tumor is composed by different groups of cells which behave differently. Some subgroups of each tumor may be more resistant to treatment than others. Some others may be more prone to progression and aggressive clinical features than others. And we were able to find that upon treatment, the subclonal structure or architecture of multiple myeloma changes in at least 2/3 of patients. Sometimes we were able to see that these changes are minimal or subtle. Some other times these changes are dramatic and correlate with resistance to treatment and different clinical features such as extramedullary spread in secondary plasma cell leukemia or malignant ascites. Looking at all the individual DNA alterations, we were able to find which genes are often driving these aggressive and chemoresistant relapses, and therefore we provide old and new targets for future therapeutic studies aiming at a better disease control and eventually cure in multiple myeloma.